Welcome back, everyone. So in this video, we are in the middle of talking about our cell cycle, and we're going to talk about some molecules that are involved in controlling the cell cycle. These are kinases, which phosphorylate and dephosphorylate, and they're controlled by cyclins. So we are going to call these cyclin-dependent kinases. So being able to phosphorylate and dephosphorylate are common ways that your cell marks things and controls them, and the cell cycle is no different. Having things phosphorylated and dephosphorylated makes a big impact on its activity in promoting movement through the cell cycle. So progression of the cell through the cell cycle is going to be driven by these cyclin-dependent kinases, or these kinases that are only going to be active when they are bound to a cyclin. The cyclin activates the cyclin-dependent kinase. During the cell cycle, the concentration of cyclins are going to rise and fall, and that means that there will be various concentrations of cyclins during different parts of the cell cycle. So those cyclin-dependent kinases will only be active at different stages of the cell cycle. So for example, mitotic cyclins, these are required for the G2 to M transition. They bind to mitotic CDKs. And so that mitotic cyclin, its concentration will go up when it's time to go into that G2 to M transition, and the mitotic CDKs then will be active. That complex, the mitotic cyclin and the mitotic CDKs, form what's called the MPF, or the mitosis promoting factor. G1 cyclins, you can imagine, they go up during G1. They're required for passage through the G1 restriction point, or that start point. And the CDKs that they bind to are called G1 CDKs. And S cyclins, which are going to rise in concentration leading up to the S phase, are cyclins required for DNA replication. There are some other kinases. There are polo-like kinases and Aurora B kinase. These also play roles in cell cycle regulation. Aurora B kinase is part of a complex of proteins that they're going to attach to this chromosomes and then to the mitotic spindle. They're at the end of cell division. So because they're kind of riding along on the chromosomes, sometimes they're called a chromosome passenger complex. You can just imagine them being like a passenger on the bus riding along the chromosomes. CDK and cyclin complexes are going to be very tightly regulated because of the role that they play in moving a cell through the cell cycle. One way that we control Cdk-cyclin activity is just how available those cyclin molecules are, just what we were talking about. But there's also a second way that we regulate them through phosphorylation and dephosphorylation of the cyclin-dependent kinase. Okay, so that first cyclin availability, you can see mitotic cyclin, its concentration rises, it is able to get to a high enough point, it progresses that cell through the G2 to M transition point, and then its level falls. Then as the cell starts going through the cell cycle again, it starts rising, leading up to the G2 to M transition, and then its level falls. And as the cyclin activity, or as the mitotic cyclin availability rises, you can see the activity of the mitosis promoting factor is going to correlate with that, because the mitosis promoting factor is only active when the cyclin is there. You can also see different kinds of cyclins, how they rise and fall. So here's the D1 cyclin, here's the D2 cyclin. You can see they cycle as a cell cycle fluctuates. The cyclins will also, their levels will cycle. Mitotic cyclins reach critical threshold here at the end of G2, which allows it to activate the mitotic promoting factor, and triggers the onset of mitosis. And then as mitosis starts getting underway, about halfway through it, those mitotic cyclins are abruptly going to be destroyed. Another way that we control CDK activity is through phosphorylation and dephosphorylation. So here, if this is your mitotic cyclin, it has to bind to the mitotic CDK for the CDK to be active. But even though that is required, it is not sufficient. Just having the mitotic CDK and cyclin complex, that is still inactive. So the ways that it is phosphorylated and dephosphorylated is pretty important for it to gain its function. So first, after the cyclin and CDK are bound, we have some inhibiting kinases that add these two phosphates to it. Those two phosphates will keep that complex inactive. There's also an activating kinase that will add a third phosphate, here this yellow one. That third phosphate is required for activity, but it won't be active until these first inhibiting kinases phosphates are removed. So then, now that we have these three phosphates, but these two are kind of blocking the active site here, it's not active, we have phosphatases which remove those two inhibiting phosphates. And now this one is active. So you have to have cyclin and CDK bind, you get some inhibiting phosphates, activating phosphates, remove the inhibiting phosphates, finally it's active. Then once you have this active mitotic Cdk-cyclin complex, it goes back in this positive feedback loop and stimulates more phosphatase activity to remove the inhibiting phosphates on other mitotic Cdk-cyclins. So you just amplify and build up the level of mitotic Cdk-cyclin complex so that that cell gets pushed through that checkpoint into mitosis. Then, now that you have this Cdk-cyclin active, it is going to start triggering mitosis by doing a lot of different things. One thing it does is it'll phosphorylate lamin. And when it phosphorylates lamin, that causes the lamina, the nuclear lamina, to break down, which means that nuclear envelope is unstable. And that is a required part of mitosis to disintegrate that nuclear envelope. Cdk-cyclin will phosphorylate condensin, and condensin will cause the chromosomes to condense, which is also a necessary part of mitosis. Cdk-cyclin will phosphorylate microtubule-associated proteins, which will then start assembly of the mitotic spindle. And they will phosphorylate different proteins in DNA repair pathways, which will kind of inhibit that messing around with trying to fix genes in DNA, so that DNA isn't opened up and getting broken. So that prevents DNA break repair during mitosis, which kind of helps protect that DNA because it's all nicely condensed during mitosis. Then, once we're getting through that cell cycle, we've gotten into mitosis, there's another important complex, the anaphase promoting complex, that will let your cell exit from mitosis. So as you build up this mitotic Cdk-cyclin, it phosphorylates something else called the anaphase promoting complex, or the APCC. When the anaphase promoting complex is activated, it is going to help that cell move into anaphase and separate the chromatids. So this anaphase promoting complex, that's this guy right here, he functions as a ubiquitin ligase. Ubiquitin is just another sort of tag. So it is going to add a ubiquitin tag to proteins. When APC adds that ubiquitin tag, it marks it as being ready to be degraded. So one of the things that APC will add this tag to is a molecule called securin. So once that securin is ubiquitinated, it has that tag on it, securin gets broken down. Securin, during other phases of the cell cycle, is bound to separase and inhibits separase. But once securin is broken down, separase is now free to go act on its own. So separase now comes up and finds these sister chromatids and will break the cohesin that is keeping them connected. So that allows in anaphase the two sister chromatids to separate. So prior to anaphase, sister chromatids are held together by cohesins. Those are the cohesive adhesive proteins. Securin maintains that attachment by inhibiting the separase protein that would degrade the cohesins. But when the anaphase promoting complex destroys securin, or marks the destruction of securin. The separase is free to go cleave cohesin and separate the sister chromatids. There's another thing the anaphase promoted complex targets. I'm going to jump back up a couple of slides. One more thing anaphase promoting complex targets is mitotic Cdk-cyclin. So now that we have gone through anaphase, we don't need to keep that cell cycle stuck at that G1 to M transition. We want to move on past it. And so the APC complex targets the mitotic cyclin for destruction. So the CDK complex activates the anaphase promoted complex, and active anaphase promoted complex breaks down the Cdk-cyclin. So once that mitotic cyclin is destroyed, mitotic CDK doesn't function anymore. That means that all of those things that mitotic CDK was doing are going to stop being done. So a lot of those changes that are of a cell exiting mitosis, so cytokinesis, chromosome decondensation, assembly of the nuclear envelope, those are all depending on mitotic cyclin being degraded, because those are all things that the mitotic CDK does in reverse, right? So by getting rid of mitotic CDK, the cell can now exit from mitosis. All right, so I think cyclins and CDKs are super interesting. You can expect that I'll talk about them a lot on quizzes and exams. So please make sure you're comfortable with that. All of that information we talked about is pretty important to this class. If you have any questions, let me know, and I'll talk to you later. Bye.